probably not many people got a confusion concepts of the HBV infection because so important in China. There's all kind of misinformation with regarding to the understanding of virology, understanding immunology, and understanding quote unquote cure. That's why this concept is so important. That's why I like to spend a little bit more time to going through all the virology. But this topic, it's really difficult because has involving three major disciplines. It's virology, there's immunology, and it's very deep in immunology. That's why I really like to hear Jun's comment. And there is the RNA-based medicine, which is Ionis and other companies' RNAi. And just quickly going through the virology, this is one of the most complicated virus among all the virus. This is what's essentially it is, it's a double-stranded DNA virus, but it's partially double-strand. Not every part of the genome double-strand. There's a portion of the circular genome is single-stranded DNA. But it's also going through RNA, so-called pregenomic RNA, involving reverse transcription. It's kind of counterintuitive because usually for a DNA virus, you don't need to do reverse transcription. For only the RNA virus, you need reverse transcription or retrovirus. But turns out in the case of HBV, it's complicated. It has to go through a pregenomic RNA, then using reverse transcription to synthesize the minor strand, non-coding strand of the DNA, then have the double-stranded DNA only in certain region. That's why the virology is already very complex. Then another complexity, unlike the mammalian eukaryotic genes, this kind of virus have overlapping open reading frame. There's overlapping gene. It's not like one segment encoding one gene. Multiple genes have overlapping. So that's created another layer of complexity because virus want to be a little bit parsimonious in using the total length of the genome, and then the overlapping genome creating some additional complexity. But regularly, we can thinking about these different RNA lengths. You know, when you run a northern blot, there's three major forms on a northern blot of RNA: 2.4 kb, and there's 2.7, 3.9 kb, and there's multiple lengths. They're encoding different protein product, which was originally defined, as people know from doing a serological test, the concept of HBs antigen, which is surface antigen. Then you have the HBc antigen, a core antigen. Then you have the HBe antigen. So these are the common antigen running through a serological test. But the life cycle of HBV is also very complex. It's going through multiple forms of genome. One of the very famous or infamous form is the cccDNA. The cccDNA stands for covalently circular closed DNA. This is very persistent, and it's very almost impossible with whatever therapeutics available to this day to eradicate, to really deplete cccDNA. You can pretty much doing the zero clearance on the HBs antigen, but it's almost very difficult. Maybe future gene editing with CRISPR may be able to clear that. But there's also the form of relaxed circular, rcDNA. Then you have also the double-stranded linear DNA. So these are the multiple forms throughout its life cycle. So the virus basically enter hepatocyte through a major high affinity binding receptor, NTCP, but also bind to heparin as a low affinity to initially arrest the virus particle. Then through high affinity receptor binding, this receptor was discovered by Dr. Wenhui Li from Beijing Institute of Life Science. That's a major discovery in 2012. This is regular a transporter, SLC10A1, but found it to be multi-purpose. It's also utilized by the virus to enter the hepatocyte. And then after entrance into the hepatocyte, you will go through multiple forms, and the linear form, the DSL DNA, eventually get integrated into some transcriptional unit. But none of the integration at the multiple sites can reassemble a virus particle. So, but they do express the surface antigen. So once the integration, especially for those patients relatively so-called inactive carrier, this can still express those HBeAg HBcAg, HBs, the surface antigen. That's why it's kind of very complex. I will have later explain a little more. Then the kind of virus particle is a nuclear capsid. It has the kind of envelope on the outside, which have the surface antigen. The surface antigen, although on the serological test, is genetically referred to as HBsAg, but it contains multiple envelope protein, like the large one, middle one, and the small one, LMS. They are all encoded in the kind of S or pre-S open reading frame. Then HBE essentially is the pre-core protein, which is an indicator of the virus replication. Then to give a kind of very quick overview about all these kind of terminology, different names, these are the LMS are the ones that's the composed, have epitopes in the HBs surface antigen. And that's the common antigen, because by expressing more and more, it will induce immune tolerance, also leading to the exhaustion of HBV-specific T cells or even HBV-specific B cells. So that's the key thing, that's the key concept. So surface antigen, HBsAg, serves as an important biomarker, stratification biomarker. You need to reduce the surface antigen in order to unleash the immune system. That's why for many therapy to work, you really need to pay a lot of attention to the amount of protein is expressed, is available in the blood. You need to reduce the load of HBsAg in order to control the tolerance or the exhaustion. Then measuring of the copy number of HBV DNA is also very important to monitor the chronic infection. And then in recent years, there's something called a new biomarker called HBcRAg. It's HBC-related antigen. It's a composite biomarker, adding together the HBcAg, HBeAg, and the P22. Together, there seems to be an interesting biomarker indicating the cccDNA transcription activity. So it's a very complex story, but I just want people to be a little bit patient. Then the discovery of HBV by Dr. Bloomberg got the 1976 Nobel Prize. It's not often for the discovery of a particular virus to get a Nobel Prize, but this particular virus, because it's way more kind of important in the study of virology, got attention. And luckily, the vaccine was developed very early through some serendipity. Interestingly, HBC, the hepatitis C virus, it's an RNA virus, never got a vaccine. So there's a different reason to explain why HCV cannot get a vaccine. But for serendipitously, HBV got a vaccine in the late 1970s and early 1980s. Essentially, people need to keep in mind, many adults in China in 30 or 40 or 50 years ago, when this virus was silently spreading, the main thing is it's a non-cytopathic virus. Even the viral load is very high, it does not cause symptoms. It can having some kind of dynamic equilibrium with hepatocyte without hurting the hepatocyte. That's why when you do an ALT/AST test, even for people with very high viral load of HBV DNA, their ALT/AST may still be normal because it's non-cytopathic. That's making this virus very silent. It's very difficult to really do the detection. That's why many of the carrier of HBV has been underdiagnosed. So when an adult, even without vaccination 50 years ago, as long as you're adult, I don't know the exact age cutoff, as long as you're adult, you have a very well developed immune system. When you encounter hepatitis B virus, it'll become acute hepatitis B, AHB. That's a self-limiting infection because immune system in adults are very well working. Usually it will contain and it will see some very transient rise of ALT/AST. Then because it's acute, eventually it will be kind of wiped out. Then even for those adults never get a vaccine, but they will be fine throughout the rest of their life because it's the self-limiting fact of this virus infection. But for young people, teenagers or younger, even neonatals, because immune system has not been well developed, unfortunately, that become a problem. It's called chronic hepatitis B. Because you will see different dynamics, hemodynamics. For example, there's a majority of the carriers of chronic hepatitis B. They will be HBe positive for a long time. It's very difficult. Only 10 or 15% of those carriers become HBe negative. So in regular Chinese jargon, those are the so-called 大山羊. Those HBe negative carrier are the 小山羊. Well, there's a major difference because those HBe negative chronic infection, the HBV DNA is relatively low, and the AST usually does not go up, and the DNA is not very actively replicating. But it's a time bomb. It can be a time bomb because there's no guarantee these viruses will stay silent forever. So you need to, that's why for those carrier, even they used to be called inactive carrier, they still need to do blood tests every year to do the to monitor the HBV DNA copy number. Just it's a longitudinal monitoring, so that anytime if the HBV DNA go up, you need to receive the standard of care therapy, the nucleotide or nucleoside analog to bring down the HBV DNA copy. Because for HBe antigen negative carrier, anytime the HBV DNA going up, their progression to cirrhosis or hepatocellular carcinoma even faster than those HBe antigen positive patients. So that's why I want the people to pay attention. It's not like these people are safe, HBe antigen negative. They need to monitor the HBV DNA copy number. The goal is to reduce HBV DNA copy number to reduce the risk of cirrhosis and HCC. Then that's the kind of, you want to keep them as infection, chronic infection, without being called chronic hepatitis B disease. But for those patients, HBe antigen positive, their viral load DNA is always high. They immediately need to go through the standard of care, the nukes or the directly acting antiviral to bring down the copy number to be similar to HBe antigen negative carriers. So that's why, but unfortunately, that's a lifelong treatment. They cannot stop taking the nukes. They have to keep inhibiting the DNA. Anytime they stop taking the pills, those viral load will rebound. Then they become very vulnerable and very dangerous. So the goal for the whole field is, can we find a way to do finite treatment without lifelong treatment? That's why the Ionis many years ago starting to using the antisense oligo and also siRNA, try to see, can we induce a long-term surface antigen loss and control the DNA even after a finite length of treatment? That's why we call functional cure. The real cure of HBV, at least as we know now, is impossible, because unless you wipe out cccDNA. That's why people should not be misled by the Chinese media, keep talking about cure. This is a quote-unquote functional cure. The definition of functional cure is you treat with some treatment regimen, maybe a combination therapy. Then after treatment, for a certain length of time, 24 weeks, 36 weeks, even several years, if your HBV DNA remain low, if your HBs antigen remain very low, then that's a very good indication. So that's the quote-unquote functional cure. And I also want to giving back a little bit more attention. I'll be very quick on this slide. It's just through the natural history of those infections, 15% of the carrier eventually will starting to have anti-HBe antibody. That's why their HBe antigen, the green line, will eventually go down. So that's good. That means they're inactive carrier, but you also need to monitor. Then the DNA copy number, only when you tune down the HBe antigen, the DNA copy number will be low. But the HBsAg remains a very important biomarker. The goal is to reduce those surface antigen to contain the immunotolerance, to contain the immunoexhaustion. That's why the goal is to bring down the surface antigen and bring down the DNA to be even lower than 1,000 international units per mil. So I won't go to too much. There's other than the 4 phase shown previously, there's also some gray zone. You can check. People can save slides, a screenshot. You can tell there's a very complex in terms of the multiple biomarkers. You need to do a quantitative surface antigen assay so that it becomes an important biomarker at the threshold of 1,000 international unit per mil or 3,000. I won't go into detail, but it's just a very important biomarker. Then for GSK, when they partnered with Ionis in 2019 for the Bepi ASO, they're thinking the Bepi can help them receiving some functional cure. Then you have to define functional cure, because otherwise, if you're vague calling it a functional cure, people don't know what's actionable, what's not actionable. So that's coming out of the kind of people's complaint that you have to take a lifelong nuke treatment. Lifelong treatment is never pleasant. So for even working towards a functional cure goal is to reducing the intake of nukes, and then ideally you will have the surface antigen loss. That's a really important kind of factor to reduce the risk of the HCC, hepatocellular carcinoma, and then try to keep DNA and the surface antigen low. So that's why you think about it's a multidimensional concept for towards a functional cure. On the one dimensional is the amount of surface antigen, which is related to immunotolerance or exhaustion of T cell and B cell. On the other dimension is the exhaustion level. Then recent years, people adding a third dimension. It becomes a multidimensional space for you to think about how do you contain that. That's the immune dysfunction. So you have to kind of triage those patients into different clinical trial. But the goal is to reducing the surface antigen so that you can somehow help the immune dysfunction. So this is why, that's why Ionis, this kind of ASO, it's a very interesting ASO. It's not just a single strand RNA, it's single strand RNA DNA hybrid. Some region of this ASO is single strand RNA with 2' modification to increase the stability. That's the methyl oxyacyl on the 2', replacing the 2' OH of the ribose. Then you also have some DNA single strand segment, which is the 2' deoxy. So those turn out to be very important to engage the viral mRNA. It's targeting RNA, it's not targeting the genome. It's targeting the RNA to, it's a common region that's shared by the overlapping open reading frame. Essentially, all the HBV RNA's expression or the mRNA amount will be contained because RNA-H, the host RNA-H, can cleave off, degrade those hybrid, and reducing the total amount of mRNA. That's why the gene expression, the mRNA level is very well controlled. But through many years of study by Ionis and GSK, they found out it's basically can work through 3 different paths. One is to reducing the viral protein, especially surface antigen. That's the most important one, with behind immune evasion and the disease chronicity. And then it reduced the viral DNA load. That's an important risk factor for HCC and the cirrhosis. Then there's a hypothesis yet to be proven is that this degradation of those mRNA or the total reduction can somehow stimulate the immune system. And via mechanisms such as the activation of the toll-like receptor 8, and I think Jun can later comment on the hypothesis of an immune system. It can somehow alleviate the immune suppression and making the immune system more competent to handle those HBV. Then in 2023 or 2022, they published a phase 2 study, which is a very important study. It's phase 2B trial called Be Clear. And it's a very complex design. Multiple groups like 3-3-3-1 randomization. Group 1 is a monotherapy for the Bepi and with a leading dose. And group 2 is different dose. The second dose is more plus the placebo. And the third dose, it's very complex. But the bottom line is that the phase 2 result, achieving an average functional cure, which is undetectable DNA, a very low surface antigen, you can see there's a negative correlation to the baseline patient. If the patient's baseline surface antigen is low, it usually responds very well. You have a much higher functional cure rate. But those percentage you have to take with a grain of salt because the total number of the phase 2B trial is only about 457 patients. But by focusing on the subgroup of HBsAg, the percentage could be a little bit tricky to assess because the smaller sample size. But the trend is very clear. The lower the baseline surface antigen, the better the functional cure percentage. On average, throughout the whole cohort, you get about 9% to 10% functional cure rate. But this trend is really encouraging, is that the lower amount of HBsAg in the baseline predict a better functional rate. That's why they want to do a phase 3 clinical trial. So that's why if you look at the detail about this, the phase 2b data, overall it's 9 to 10%, but the subgroup is very interesting in terms of the participants achieving functional cure. So the design detail, I don't need to go through all that because the data has not been fully presented. GSK and Ionis only released a press release on January 7th this year, prior to the JP Morgan conference. But the bottom line is there will be a major presentation at the European ESSL Congress around late May. So that by that time, they will fully present both the BL1 and the BL2 phase 3 data. But from the top line highlights from the press release, you can already tell they're very confident because the phase 2b observation of the low HBsAg subgroup giving a very high percentage. That has been confirmed by a phase 3 trial. And this phase 3 trial having a lot of, almost 2,000 patients across 29 countries. That's a very full scale. And mainly the primary endpoint is you compare the functional cure rate as defined previously, and you just having a very sustained loss of surface antigen and not detectable DNA 24 weeks after completion of a finite course treatment. Usually you can do weekly injection of Bepi for 6 months, then even 24 weeks after stoppage of the treatment, you can still have some maintenance of functional cure rate. Then they compare Bepi plus SOC, typically is the nuke, the nucleus analog, versus the SOC alone. They have a significant higher functional cure rate in the Bepi plus SOC group. So the exact number, you can get a glimpse from CSO Tony Wood's kind of Q&A at the JP Morgan conference transcript, when he mentioned that they are optimistic you can average get a 15 to 20% functional cure rate in the B-well kind of population group, which is better than the 9 to 10% in the phase 2b trial, because now they have a full-scale phase 3 trial. So that's very encouraging. So overall, you can say the takeaway is that this phase 3 success really proving that off-therapy HBs surface antigen loss and the virus suppression can really be achieved on a phase 3 trial, which is a registration trial. So GSK will file for NDA by the end of first quarter 2026, so that now people can finally, after this major progress, we can talk about a functional cure rather than just talking about theoretical cure. And this also shows that RNA-directed surface antigen suppression is a clinical actionable pillar. It's not everything. It's a component of a combination therapy strategy, but it's an important cornerstone for the future. Any future kind of functional cure therapy may have to involving RNA-directed surface antigen suppression. Then to even go better, can we do better than 15 to 20%? And that's my next several slides will talk about that topic. But it's already showing that a stratification biomarker for surface antigen, this is very important. Without this biomarker, it's very difficult for this phase 3 trial to get a statistical significance. So now I'm going to, we can wait until late May or early June to talk about the detailed data, but I can say that GSK already envisioned what's next. After this phase 3 trial success, you get FDA approval. Can we do even better? So that's why, because ASO can enter the nucleus, but ASO has to deal with varying amount of the total RNA expressed by HBV. Some patients, if the RNA is really very abundant, no matter how you change the dose, those relatively smaller amount of ASO has to deal with a larger amount of the RNA. So one of the idea is to, can we treat with siRNA, combine with ASO? siRNA, longer half-life, can deal with RNA suppression even longer. But siRNA, there's a debate whether siRNA can enter the nucleus. So ASO can surely enter the nucleus along with some other chemical small molecule compound like PAPD 5/7 inhibitor can further reduce the stabilization of mechanism. So that's why we can see that there's a potential for siRNA to combine with ASO. Then Jun can comment about immunomodulation regarding toll-like receptor, PD-1 checkpoint inhibitors, as well as the targeted immunotherapy. But I also want to get people's attention. This space is picking up momentum. More and more small molecule compound, new modality are entered the clinical trial. There's capsid modulators, siRNA, antisense, which will be the bottom line for GSK Ionis. But there's also other molecules enter the clinical trial. So combination therapy will be the theme eventually if we really to do a high rate, higher than 30% functional cure rate. This is from some analysts, Wall Street analysts, they really highlight what's upcoming for GSK and other company. So yeah, I won't stop here and turn over to Jun for additional dive into the immunology.
All right, so thanks, Ethan. That was already great, and it covers most of the topics. So basically, I was actually studying HBV back to 20 years ago. As you know, when I was in Chinese Chemical Sciences, when I was a PhD student, we are thinking about how we can handle the HBV issue. We have such a great population. Many of those has been infected by HBV. So the major problem in the whole population, and for a long time, there's not too much solution. I think for HBV, the major problem was, as you know, so many patients get infected, and those are chronic infection. And one issue is those patients will be almost lifelong infection. Many of those are from the mother to baby, that kind of infections. And then one issue is those patients will become chronically infected and will lead to chronic liver disease, can be chronic hepatitis, and even cirrhosis, fibrosis, and ultimately leads to cancer development. So that is very lethal, can be very lethal disease. But once patient get to the fibrosis or cirrhosis or cancer stage, then you will be very severe. So that's why for a long time, people has been thinking about more focusing on antiviral to prevent. Of course, vaccine has been very successful, but once people are already chronic infected, vaccine is not effective. So then we're talking about how we can use some strategies to target viruses to shut down viral load. So that's what Ethan has been talking about, GSK data, ASO, and even I'm going to talk about, as I showed here, Roche data. So Roche actually already have tried, siRNA, and before siRNA and ASO targeting HBV directly, before there was some kind of nucleotide analog therapies. It's called NA, nucleotide analog. So that's one of the typical therapies. And even before that, there are some kind of immunomodulators like CpG, toll receptors. Actually, PD-1 trial was already tried back to 15 or 16 years ago for HCV and some HBV trials. But the problem was, okay, the idea was to boost immune system to especially T cell responses to antiviral. So actually, early trial showing some reduction of the viral titer, but the problem was T cell at the same time might cause more liver injuries. So that's why actually the PD-1 trial has been already tested in some of the virus HBV, HCV trials, but the toxicity has been problematic. Even those type of trial can be before PD-1 trial in cancer or around the same time. So for 20 years ago, people have been trying immunotherapy, but later time people found maybe targeting viruses can be more straightforward. That's why ASO and siRNA coming back. And I think the data is getting quite interesting, especially as Ethan mentioned, the ASL data can lead to 15% or 20% of HBsAg conversion. Mostly, and those type of therapy have a biomarker. If you have a lower antigen amount, surface antigen amount, you have a better response. If you have a higher titer, you have a weaker response, which is interesting, right? But I want to point it out through those two slides that the data is not great enough. So this data is SRA trial, siRNA trial, published 1.5 years ago, 2024. So basically, Roche have tried several groups, siRNA alone, different dose. They can reach to 7% or something response. But they also tried another two, which is the green bar is toll acceptor TLR agonist trial. So if you have a combination with immunotherapy, you can switch from several percent to 12%. And also, if you want to combine with pegylation type interferon is also a therapy. As you know, type interferon is very antiviral. So type interferon therapy has been widely used for HBV, especially in China, because China have a huge population. So in this trial, Roche found siRNA HBV combined with type interferon, pegylated type interferon. It's really effective. Can be 20% versus 3 or 7% of monotherapy. It's great data. But the problem was, if you look at this blue bars, the safety. This is grade 3 or 4 safety adverse event was from 10 something to 50%. So basically, the response has been greatly lifted, but the safety is really the problem. So that's why Roche, although they see some signal from the single agent of siRNA, but they think several percent is not good enough. They would like to see something like, like bigger number, but unfortunately, those compilation trial, also bring more toxicity. That's why pegylation interferon combo, I think, is very effective but toxic. That's why Roche later time discontinued most of the programs. So beyond what Ethan has described, and actually GSK also have done the pegylated type interferon combined with ASO, as I showed here, the big together trial. So Ethan just mentioned 15% or 20% by ASO alone, but the combination with pegylated interferon almost give you about 20 or 30 or 40%. And this type of data can cross many other combinations. For example, siRNA combined with pegylated interferon or nucleotide analog pegylation. So in general, combination with pegylated type interferon is very effective, but the major problem is toxicity, as showed by the Roche data. One slide more, thank you. So this is the data actually ongoing trial with GSK. So Yusen just showed the BWEL1 and BWEL2. Those two trials are, the ASO trial combined with a nucleotide analog. Nucleotide right now is more like standard. So personally, I think the Beat Together trial and with pegylated type interferon, that is a trial we really have to take a look whether safety is better with SL compared to siRNA. That is something we didn't know. So if the data is very similar to Roche data, I would say this might be problematic as well. So maybe I agree with Ethan that the BWEL1 and 2 trial were going to be approved very soon. But the problem is exciting, of course, it can be the basis for the further compilations. But if the pegylated type interferon is interesting to get better efficacy and the safety is manageable, not that problematic as Roche, I would say big together trial might be more interesting. But we don't know, right? Prediction is still toxic, but whether it's more toxic or less toxic compared to Roche trials. So in general, I think for the unmet medical needs right now is I do believe GSK will get approved for those trial, especially combined with a nucleotide analog. But the problem is only 10% or 20% of response, and also only in low antigen related patients. So we do need more therapies, especially safer interferon-based therapies. And as I mentioned, other immunotherapy options, for example, PD-1 combo, it could be interesting, right? before people treat with very heavily treated with PD-1, maybe too toxic, maybe combined with ASO, we can use a lower dose of PD-1 or something else. We didn't know, but that's something definitely worthwhile to be pursued. I do believe new doors, are open, widely open. The ASO can be the backbone, and even siRNA can be potentially useful. But for the ultimate cure, we do need a immunotherapy. For the more patient to get responded, we need immunotherapy. So that's why this is also a review paper, commentary paper, from Gut. It's a very very good journal for immunotherapy for viral related liver disease. So they are talking about pegylated type interferon is back because before it's too toxic. Before without ASO, you have to treat very heavily, too toxic. Outside the US, sorry, outside of China, not too much patient has been treated with pegylated type interferon. But with this, it could be possible. And again, safer, maybe there are some other approaches to modulate type interferon can be more useful, more interesting, and more potentially maybe like combined with PD-1, but it has to be much lower dose. Those type of stuff, we're going to change the landscape on top of ASO, maybe also on top of siRNA. That is basically my comments. If you have any comment, if anyone have any further comments, we can discuss. Thank you.